Perfect, thank you. All right, everyone, thank you so much for joining us. We will now have a presentation by Dr. Peter Harrison. He is a professor from the Faculty of Sciences and Engineering from the Southern Cross University. He has vast experience in the topics that he is about to discuss, and the name of his presentation is Optimizing Reef-Based Coral Larval Restoration and Collaboration in Honduras. And we will have a question and answer session after his presentation that you will all be able to be a part of. So without further ado, Dr. Peter, the floor is yours.
Thank you. Hello and welcome to this talk, and thank you to everybody who's been involved in enabling Alexandra to get to the Honduras to talk to you, and also for organizing this video link for my talk this afternoon and evening in England, where I currently am at the moment, visiting some colleagues in Bristol. So what I'd like to talk to you about today is the coral reef crisis and what we can do about it. What we all are very aware of is that the ongoing loss of foundation reef corals, the corals that create the entire reef structure, is happening at a pace that's so much faster around the world than natural recovery is allowing those damaged reefs to recover. And therefore, we have a global coral reef crisis. Oh, hang on. For some reason, I can't get, there we go. So what we urgently need is effective and scalable coral restoration methods that can be applied to a wide range of different reef environments, while we all continue to push hard for global action on reducing greenhouse gas emissions and climate change impacts. Coral larval restoration aims to rapidly restore resilient, self-perpetuating breeding reef coral populations using larval restoration techniques based around natural sexual reproduction cycles of corals in order to catalyze much larger scale coral recovery and therefore reef recovery once the corals are restored. We're all very aware that reef-building corals are absolutely essential for healthy reefs, and we know that reefs have immense ecological, socio-economic, and cultural values. We're also mostly aware that sexual reproduction is essential for population recovery, for reef resilience, and adaptive evolution, which allows corals and other organisms to adapt quickly to changing climates and changing reef environments. So sexual reproduction is fundamental and absolutely critical to the restoration of corals on our damaged reef systems. We also know that strong larval supply, which occurs from healthy coral environments, is essential for producing millions or billions of larvae for settlement, recruitment, and ultimately coral recovery. During the larval phase, they can also disperse, and therefore they allow for genetic exchange over large distances and connecting different physically separated reef environments. So sexual reproduction cycles are extremely important for recovery and interconnectivity of different reef systems. Sexual reproduction is important for increasing genetic diversity and allowing for evolution, adaptation, and resilience. And around the world, we know about the reproductive cycles of more than 400 coral species. The vast majority of the species that have been studied so far, and about 85%, are what we call broadcast spawners. So if we look at their life cycle, once the corals get to sexual reproductive maturity at a certain size and age, they grow eggs and sperm in each of their polyps. They release the eggs and sperm during spawning events, and masses of the eggs and sperm collect at the sea surface, where the egg-sperm bundles can break apart and allow for cross-fertilization of eggs and sperm from different colonies of the same species. And sometimes also hybridization occurs, where eggs and sperm from different species can be compatible and you end up with hybrid larvae. Once that fertilization has occurred, the embryos undergo fairly rapid cell division over a period of about 24 hours until they develop back up into a little ball, at which stage it starts to grow hair-like cilia and becomes a larva. Those larvae develop over a minimum of 4, 5 to 7 or 8 days. And therefore, during that period, many of the larvae actually drift away from their parent reefs. Some of those larvae drift back onto the same reef. Some of them drift away onto other reefs. But in many cases, many of those larvae end up drifting out to sea, and in some cases, never are able to reconnect with another reef environment, and therefore they're lost from the system. So the way in which corals cope with this inevitable loss during this phase is to produce so many eggs and sperm that they produce huge numbers of larvae so that the loss of some of those larvae is not critical, and therefore their reproductive cycle can still be effective. So during this period of dispersal from the parent reef across to other reef systems, you end up enabling some genetic exchange between the populations. And once the larvae become competent to settle, they actively swim down through the water column and start to search for somewhere suitable to settle. That choice of settlement site is really important, because once the planular larva settles and grows into a juvenile polyp, that's where the adult coral will have to live and survive. And so the choice of settlement site of these larvae is critically important. Once they have settled, they metamorphose from this elongate larval form through into a little polyp. And after about 36 to 48 hours, as in this photo here, you can see the little white skeleton has started to develop and the tentacle buds around the mouth to allow the polyp to start feeding on plankton at night. At this stage, most of the settlers die because they run out of energy, or they get eaten by predators, or they've ended up settling in the wrong environment, and therefore they just don't survive to grow into larger corals. But for those that are really well adapted to the reef environment, they can thrive, and once they get to a visible size, the rate of mortality greatly reduces, and therefore they can become very fast growing and replenish the degraded populations very quickly. So at this stage here, they're microscopic, about 1.5 to 2 millimeters maximum. By 2 years of age, they're growing into a small plate size, and by 3 years, they can become very large plate sizes and be sexually reproductive, therefore closing the life cycle. The other groups of corals, about 15% of the total coral species that have been studied so far, have a slightly different form of sexual reproduction, where sperm are drawn into the polyps and fertilize the eggs internally. The larvae develop within the polyps, and when they're released, they rapidly settle, so they can settle quite close to the parent colony or drift away to nearby reefs. But once the larvae are released, they go through the cycle of settlement, metamorphosis, survival, and growth through to a reproductive adult, and their life cycle is closed. The global coral reef crisis is caused by reef resilience being overwhelmed by chronic human impacts coupled with severe natural disturbances. And as a result of that, not only do we lose living corals from these reef systems, but we lose breeding corals. And that's really important, because it's the breeding corals that are fundamentally important to the recovery of reef systems. As a consequence of so much loss of breeding corals, we get impaired or no larval production, and therefore very low or no natural recruitment. The causes of this are very obvious to most people. Climate change is one of the biggest threats to reefs around the world, causing marine heat waves and mass coral bleaching. We also have increasing severity and frequency of massive storms, cyclones in the southern hemisphere and typhoons in the Caribbean. Crown of thorns and other predators eat corals. All sorts of human pollutants damage corals and stress them, causing them to die. And In some areas of the world, and particularly in Southeast Asia still, blast fishing is still occurring and unregulated overfishing is occurring. And therefore, the loss of the fish component on these reef systems further damages them. So what we need to do is urgently restore coral communities to kickstart the recovery of damaged corals while we act on global climate change and ocean changes to support the reduction in greenhouse gas emissions. In the most recent global analysis of the state of the world's reefs, it's estimated that more than 50% of the world's reefs have already been lost, and many other reef systems are rapidly declining throughout most reef regions around the world. In the Caribbean, the same process is occurring. Mass coral bleaching events, outbreaks of diseases threaten the very reef existence. And in the recent workshop I was at in the United States earlier this year, it was very obvious that of the 3 major reef builders that are occurring on the Florida Reef Tract, there's been no natural recruitment of any of those species along the 300 miles of the Florida Reef Tract for the last 2 decades. So essentially, those reef systems and those communities are functionally extinct. Healthy reefs have high coral cover, resilience, and diversity. Degraded reefs simply may not recover unless sufficient larvae are pumped back into the system to enable that sexual reproductive cycle to occur again. Even on the Great Barrier Reef, which is the world's largest reef systems, we're seeing dramatic losses of corals. And even though there was quite recent recovery and very high percentage cover on some of those reefs, the most recent mass bleaching event, which is still unfolding on the Great Barrier Reef, has pretty well wiped out all of the advances we've seen in the last 5 years. So over recent decades, we've seen a 28% loss in coral cover, in mean coral cover. As a result of that, a doubling of dead coral areas that are now covered with macroalgae. So the conclusion is that although passive reef management is incredibly important for maintaining many of the reef functions, it's simply overwhelmed by the scale of the impacts that human activities are causing. And therefore, by itself, it's not going to be adequate to create the processes that allow these reef systems to recover. And therefore, we need to be thinking about active interventions and coral reef restoration. This is incredibly urgent. As you're all aware, NOAA recently declared the 4th global mass coral bleaching event, indicating that marine heat waves and high probability of mass bleaching events were likely to occur in every ocean basin and in most of the reef systems around the world. That just highlights the urgency of the need for effective restoration on reef systems. So the key question is, if we're going to intervene, when do we do it? Where do we do it? And how do we do it so that we can rapidly recover corals and reef health? As I'm sure you're aware, and I think many of you are probably already heavily involved in coral gardening approaches with asexual fragmentation. Around the world, there are literally hundreds of these projects going on. It's the most common method. It's relatively simple to use, relatively simple to train people with. And it can be very effective at creating small-scale increases in coral biomass, which is good. But the fundamental problem with asexual fragmentation and coral gardening is that it has very limited genetic diversity. And as a consequence of that limited genetic diversity, it's got low resilience to climate shocks, such as marine heat waves and mass coral bleaching. And unfortunately, around the world, we've seen many instances of beautiful outcomes from coral gardening being wiped out by the next major bleaching event. In some very careful modeling analysis that was done, looking at the most effective way to restore reefs at very large scale on the Great Barrier Reef, and it also has global application, the conclusion has been that this approach, the most common approach, which is asexual fragmentation and coral gardening, is not going to be cost effective for very large scale coral restoration. The alternative approach is sexual-based coral restoration and the production of many millions or billions of larvae. It's relatively low cost, but has the consequence of very high early mortality, which is a natural part of the reproductive cycle of corals. The big advantage it has here is not only an initial lower cost, but also massively increased genetic diversity and therefore higher resilience capability and evolutionary adaptation. There are a range of rapidly emerging methods, but overall it's considered that this is most likely the best scalable solution for large-scale restoration in the future. I was involved with the discovery of mass coral spawning on the Great Barrier Reef in the early 1980s. And what was immediately obvious when we started seeing these incredible mass spawning events was that the coincident spawning of so many colonies from the same species and so many species altogether created access to huge coral spawn slicks that contain billions or trillions of eggs and developing embryos. That discovery revolutionized and changed our understanding of sexual reproduction around the world. And it also now enables the production of millions of genetically diverse larvae for restoration and increasing resilience. And that's the work that I've been focused on for the last 10 to 15 years. The coral larval restoration concept was something that I thought about way back in the 1980s, shortly after we had discovered the mass spawning and then had the first major bleaching event on the Great Barrier Reef. And so the evolution of the larval methods and the equipment are detailed in a technical report that I wrote with a colleague, Dexter de la Cruz, and published a couple of years ago. And Alex will be able to give you access to those particular papers and technical reports if you need them. So how do we go about larval-based restoration? Well, essentially, it's quite a simple process. It consists of 5 phases, and it follows the natural sexual reproductive cycle that we talked about just previously. The first thing we need to do is locate healthy breeding corals on what we call a source reef. And if that reef has been impacted recently by extreme heat waves and mass coral bleaching, then you know that the surviving corals that are still present on that reef have higher heat tolerance than the ones that died. And unfortunately, that very rapid and very important selective filter is occurring pretty much on most reefs around the world and in many reefs right now, including the Great Barrier Reef. So the surviving corals are going to be more heat tolerant. And we do know from some recent work that's been done by colleagues that the heat-tolerant adults produce gametes that produce larvae that are also more heat-tolerant. So that genetic heat tolerance basis is passed from one generation to the next, which gives us hope that by breeding from surviving corals, we can therefore restore more resilient populations on these damaged reefs. We go around and do some rapid surveys to check how many corals are spawning, and then usually do some baseline surveys to establish what the health status of the reef is prior to our interventions. We then collect the spawn during the spawning periods, and we can do that even if it's just a really light spawn with just 1 or 2 species spawning. But when you get really big spawning events, it becomes very simple because you can simply lean over the side of a boat and pick out the spawn with buckets and collect millions of eggs and developing embryos in a very short period of time. So that's the second phase, the spawn collection. We then transfer the spawn into some specially developed floating larval pools that I've been working on and developing for about the last 5 years. These are a very simple process and a very effective way to raise tens of millions of larvae. And in the image here, you can see 2 of the pools floating in the Great Barrier Reef, and all of that area in there is covered with coral spawn. So you can routinely raise about 5 to 10 million larvae in each of these 4 by 4 meter floating larval pools. Once the larvae have developed to the point where they're desperate to settle after about 5 to 7 days, we can either settle them onto bits of dead coral or onto limestone tiles or other manufactured devices, or we can free release them back onto the degraded reef systems. And then once we've done those releases and transferred either the settled polyps on these devices or tiles back onto the reef, or the free release of larvae, we then start monitoring the settlement rates, and then over time, monitor the development and survival of those corals through to reproductive sizes and beyond. So just to show you visually these 5 stages, we've done lots of coral larval restoration experiments at relatively small scales in many reef trials in a range of different areas in different parts of the world. But what we need now to do is to scale up this larval production so that instead of producing routinely tens of millions of larvae, we produce hundreds of millions of larvae or billions of larvae over the next few years. And therefore, we can massively increase the supply of larvae for larger scale restoration. So as I said, the first phase is to locate healthy breeding corals. So we can do that by simply doing a rapid gravid coral check, looking in broken sections for pigmented eggs, which we know mean that those corals are going to spawn around the next full moon period or sometimes around the next new moon period within that lunar cycle. Once the corals are spawning, then we can collect the spawn slicks, which contain billions of eggs. So collecting coral spawn is planned to increase the numbers of larvae we can produce, but also to maximize cross-fertilization of the eggs and sperm from the corals we're collecting from. And therefore, by doing that, we're massively increasing genetic diversity in the larvae and therefore allowing for the possibility of some of those genetic combinations to better match the changed reef environments that we're now facing. So during coral spawn slick collection, even if it's fairly light, we can use a spawn skimmer at the surface. As it gets more and more dense, we can use nets, and this is basically just a pool scoop net that you use in a swimming pool modified with a plankton net at the base and a small collecting cone. We can also put spawn cones over the top of the colonies and collect the eggs and sperm bundles that rise up to the top of the containers here, and then bring them back to the vessel or back to the laboratory, combine them, let fertilization occur, and then massive larval development. One of the most effective ways that I've developed in recent years is what I'm calling a spawn catcher. So we basically have a modified larval collection pool, and it has booms out one side and an opening closing end to the net. And you can see in this image here, that was just passively collected. So as the corals were spawning just up current from where we had anchored the spawn catcher, millions of eggs are accumulating. And that particular image there at the surface of it shows about 150 million eggs accumulating in that spawn catcher. So there's a range of different ways that we can go about collecting the spawn directly on spawning corals on the reef. We can then rear those larvae in mass larval culture pools on nets. So we're now routinely culturing tens of millions of larvae to more than 150 million larvae in some of the recent projects. We started this work in the Philippines, funded by ACR and DFAT grants. And then I took some of that information back to the Great Barrier Reef and developed a more effective pool system, which is the inflatable larval pool. And we're now bringing that back into the Philippines, to the Maldives, and hopefully to the Honduras in the future. As you can see from this image here, this is a 4 by 4 meter inflatable pool system. And then within that, we have a 3 by 3 meter net. The upper part of it is created from vinyl. Then we have fine plankton mesh net through the middle here to allow for seawater to float through the net system while containing the eggs and sperm and the developing larvae. And then at the bottom of the pool, we have a tube, which is rolled up during culture, but when these larvae are ready to settle, you can simply unroll that tube and release the larvae, or attach a pipe to it, or you can draw the whole net up and collect all the larvae in here and transfer them to some other means to deliver them onto the reef. This is an image of the same types of larval pools that we're now deploying in the Maldives. This image here shows the largest of the larval pools that I've created so far. It's a 13-meter mass larval culture pool, and it's capable of raising more than 200 million larvae just within that one system. So you can scale this process of larval production either by using many of these smaller pools or gradually increasing to larger pool size, depending upon what sort of vessels you have, how many people are ready to work on this process, and how much spawn you're likely to collect. The fourth phase is the delivery of those larvae onto degraded areas, or settling them onto a range of devices or on natural limestone tiles, which we're doing substantial work on now on the Great Barrier Reef. So you can either release the larvae under net systems to concentrate them during settlement. You can attach a pipe to the base of the net on the floating pool and therefore squirt the larvae onto the damaged reef systems. Or you can use a video showing one of the larval bots that we've been using. So this is one of the robots that a colleague of mine, Professor Matt Dunbabin, has been developing. And it's a GPS-controlled robot. So you basically set its coordinates and it goes off over the reef, delivering larvae in the target reef areas. So there's a range of different ways and very adaptable ways that we can use the larvae that we've created to either settle them before we put them on the reef system or releasing them directly onto damaged reefs. We then monitor the outcomes to look for how many larvae settle, how many of the settled larvae survive and grow over the next few months. And then over longer term, we're looking for changes in coral cover, changes in fish assemblages in the larval restoration sites compared with the control sites, which have none of the cultured larvae added to them. Ideally, we keep monitoring those sites until we've proven that the restored corals are now sexually reproductive and therefore creating larvae that can be dispersed onto other areas of the reef that have been damaged, and therefore increasing the scale and the impact of the system. I'll now just quickly run through a few examples from some of the Philippines work we've been doing, funded through ACR and the Australian government through Department of Foreign Affairs and Trade, and also some of the work that I've been doing on the Great Barrier Reef. So like many places around the world, the Philippines used to have fantastic reef systems. And in fact, it's the center of marine biodiversity within the Coral Triangle, together with Indonesia. But unfortunately, blast fishing, typhoons, and a range of other human impacts and pollutants have destroyed many of the reef systems. So instead of being covered with beautiful corals and diverse fish, they now look like the image here, which is mostly dead coral surfaces covered with algae. But many of those badly degraded reefs have potentially good recovery potential. They just lack larvae. So we did some experiments with one of my PhD students at the time, Dexter De La Cruz, who's now a postdoctoral researcher working with me. And what we did was look to see whether or not simply adding larvae back into these degraded reef systems would enable any recovery. There have now been a whole range of different successful reef trials which have shown that we can re-establish new breeding coral populations within 2 to 3 years, and we're now transferring that knowledge through capacity building and knowledge transfer and stakeholder training into 5 regions of the Philippines and expanding the scales of the operation and the impact. During Dexter's PhD, he did an experiment where he was releasing larvae into a series of controlled plots, and therefore was able to control the rate at which the larvae were able to settle, and then comparing them with natural areas on the reef that were not restored with the larvae. And not surprisingly, we got very dramatic changes in the larval restoration plots, very high rates of larval settlement, and rapid recovery of the reef system. So you can see here after 3 years, all of these colonies here, this was previously entirely devoid of corals, and all of these colonies here have grown from larvae that were released during Dexter's first experiment. And we published that a few years ago in scientific reports. And again, we can provide you with copies of those papers if you're interested. So that was fantastic, because what it showed was that the idea that I'd had many decades previously, that by simply adding more larvae into degraded reef systems would enable some recovery, showed that that could be done. So we then challenged the system and said, well, if we tripled the amount of larvae that we put into these degraded reef plots, would we get 3 times as many corals out? And the short answer is yes, that's exactly what we did. So a few years later, I led another Philippines-based restoration with the same species, and we used the spawn from those restored corals plus some wild spawn and tripled the numbers of larvae that were put onto the degraded reef areas. And after 3 years, as you can see here, even more breeding corals were present. The other exciting thing that we found was that after 2 years of growth, some of these corals were actually large enough to be sexually reproductive. And we hadn't even realized that corals could grow to sexual reproduction size within 2 years. So that was a really exciting outcome, because we've closed the cycle on these highly degraded reef systems and restored breeding corals within 2 to 3 years. Those increased fish habitats have attracted more fish into the system. So as we monitor the fish communities, we're seeing increases in the diversity and numbers of fish in the restored reef areas compared with the degraded control areas. I've done some similar work on the Great Barrier Reef, supported by the Great Barrier Reef Foundation. So back in 2016, we did a similar experiment where I used 3 different densities of larvae and a control, which had no extra larvae added into it. And not surprisingly, as we increased the number of larvae in the experimental plots, we got increased larval settlement and increased numbers of adult corals on each of those plots. So in the high density areas, we ended up with much higher numbers of corals than in the medium larval density, and the low larval density was like the controls. There weren't enough larvae provided to create a significant impact. So from that, we know that the more larvae you put into the system, the higher the rate of coral restoration can occur. And on the Southern Great Barrier Reef, we were producing sexually reproductive corals in this slightly slower growing species at 5 years. The most impressive outcome we've had so far is again funded through ACR and the Australian government in the Philippines. So the Philippines team, we started working in the 100 Islands National Park with highly degraded reef systems. These had been blast fished, they had been smashed by typhoons, and a range of other human impacts. So you can see that essentially you've just got reef rubble, very few corals, and some macroalgae present. In 2020, the team reared about 18 million larvae, and it was just at the outbreak of COVID, and they were ordered to go back into a safe environment in the laboratory rather than being out on the reef system. So they mass released those larvae onto this degraded reef area, and this is what's occurred 3 years later. Here you can see proof of the concept that by producing and capturing coral spawn with a range of different species present, putting those larvae into protective larval pools to rear them, and then releasing those larvae onto the degraded reefs can result in reef restoration of many species over a rapid timescale of just a few years. And equally exciting was the fact that nearly all of these coral species were sexually reproductive and mass spawning after 3 years. So just to show you that comparison again, that's what the system looked like before. 3 years later, we have a diverse, functioning, healthy reef, which is attracting breeding size fish into these restored reef areas. So to conclude, we know that mass larval restoration can be successful even on highly degraded reef systems. And many experiments have now shown that we can produce diverse breeding populations of restored corals within 2 to 3 years. But we need to scale this process up and continually improve the methods and make them simpler and adaptable to different people using them in different reef systems. So that's why we're focused now on building regional stakeholder networks with different groups working throughout the Great Barrier Reef in 5 regions of the Philippines. And most recently, I've started working again in the Maldives with a CORDAP-funded project, and we're working with stakeholders from 4 different atoll regions to develop this process there. We're selectively breeding from heat-tolerant corals because they're the only ones that are surviving on these reefs now, and therefore that heat tolerance will be passed on to their larvae and therefore their recruits to increase the resilience of the restored populations. We're in the process of now working out how to scale this process up from smaller experimental plots to multi-hectare areas, and hopefully in the future into much larger reef areas. And we know that not only is there benefit from restoring corals for the reef system and the ecology, but it has important socioeconomic and community benefits through increased production of food for fisheries and increased income to local communities through enhanced tourism. So the conclusion is that this approach of sexual-based larval production and larval restoration is suitable for restoring corals on most reef systems. And that's why we're focused on trying to engage with as many people as possible to explain how we go about it and what the benefits can be of using this technique while potentially continuing on with the fragmentation and coral gardening approach as well. Okay, I'll just stop sharing my screen now, and I'm very happy to answer any questions that anyone has.
Thank you so much for that presentation. My question for you is that we have some challenges here in the Caribbean because the majority of our corals are more massive and boulder-like, and it's much slower growing than the acroporids and the bacilloporids and the branching corals that you have in the Pacific. So, to have the larval success here is great, but to get the kind of recovery you're seeing in the Philippines and the Great Barrier Reef in 3 to 5 years, realistically, it would still take 25 years maybe to start seeing some of these massive corals get to reproductive age, which is not really the time scale we can afford to rely on. So I'm just wondering if those considerations have been thought of your end or over here.
Yeah. Yeah, look, it's a really good point. I'm very aware the Caribbean is very different from Indo-Pacific reefs. I've been talking with colleagues in a range of different areas in the Caribbean, and so we will be trialing some of this approach very soon. But we do know that even slow-growing massive corals can be sexually reproductive within 5 to 8 years. And the point I'd make is that it's about scaling up the production. So using and collecting the spawn from as many of those surviving sexually reproductive corals as possible, increasing genetic diversity by combining the eggs and sperm to allow that enhanced resilience to occur in the restored population, by increasing that larval production, which is still possible even with decimated populations. We've been doing that in different areas around the world, and we've been using brain corals as well. So the fact that you've lost most of your acropora corals, which were really important reef builders in the Caribbean context, doesn't deter me from thinking that it is possible. It is really about trying to maximize, capture as much spawn as you can from the remaining corals that you do have, and allowing for the fact that brain corals are going to be slower to reach sexual reproductive maturity. But by increasing the larval supply of those corals, you will still get significant outcomes through the growth of those corals within that to reproductive size within 5 to 8 years. And like I think everyone in the Caribbean, nobody's completely given up on trying to save the critically endangered acropora populations as well, because those things are, massively important. They used to be historically incredibly important and ecologically really significant in many of the reef systems. And therefore, so much effort is going into working out how to start applying these sorts of processes into those remnant populations of acropora as well. So I think there is hope for the future with using some of those faster growing corals as well.
Thank you, Dr. Peter. Do we have any other questions?
Yep.
Hi, I'm Velvet. I'm a dive instructor here in Rotterdam. My question was more like you talked about the recovery of stony corals. What about soft corals? Do they get the benefit on this restoration?
Yep.
You do spawning too, or they just go in a collateral benefit of the intervention? Thank you so much.
Yeah, no, that's a really good question. Yes, we do know that soft corals can, have very similar reproductive cycles to the hard corals. So the stony corals are what I focus on because they're the reef builders, whereas the soft corals don't contribute significantly. Most soft corals don't contribute as significantly to reef building. They're really important in the reef environment, but they're also major competitors for hard corals. We know that most soft corals have chemicals in them that are toxic to hard corals, and therefore they're in competition with hard corals. So on the Great Barrier Reef, for example, we definitely select against collecting soft coral spawn. If it's mixed up with hard coral spawn, we ignore it and go on to the next patch, which is all hard coral spawn. But we do know that within our cultures, sometimes we do get soft coral larvae developing. And for us, that's not a desired outcome because we want to focus on, restoring the reef builders rather than the soft corals. But these processes can definitely be used for soft corals as well. And I'm sure that different types of gorgonians and many other species can be used. And in talking to colleagues, some people are interested in using these larval pools for production of sea urchins, for mollusks, and even for reef fish. So I'm talking here with a colleague, Professor Steve Simpson, who's very well known as the pioneer of the acoustic attraction processes for settling reef fish. And we're talking about using these larval pools to attract pre-settlement reef fish in and then protect them in the larval pools using a giant light trap at night, and then allowing them to grow in safety for a couple of days and then releasing them at night back onto the reef when they're more likely to survive, because we know that the mortality rate of those settling fish is extremely high as they come back into the reef system. So this approach can be used for soft corals, in short answer to your question, but it can also be applied to a whole range of other spawning groups or those that produce planktonic versions, like even reef fish.
Thank you so much, Dr. Peter. Do you have any more questions? Dr. Harrison Besiaspa, Executive Director of Rotterdam Marine Park.
Right.
very happy to have you here and having this opportunity to talk to you and have you virtually. I know you couldn't be here personally, but Alexandra has been giving us all the efforts you've been doing in the Great Barrier Reef and also elsewhere. And I'm happy to tell you that we are colleagues in terms of our CORDAP and your CORDAP also project. So we will be sharing uh, our own projects information with you and all the other CORDAP projects in the world. I would like to ask you, one of the main challenges that we have is how can we scale up our sexual restoration efforts? How do you think which will be the three or four main obstacles and opportunities we can get in order to scale up our project and our restoration program here in the Caribbean?
Yeah, thank you. I guess it's a little hard for me, and I do apologize that I couldn't get over to be with you like we'd originally planned. But the timing, I had to book this study leave process through the university last year, and therefore the timing couldn't work. But it's wonderful that Alex has been there and been able to share with you the work that she's doing, which is really pioneering work on coralline algae, which is also really important and directly applicable to restoration in the Honduras reefs as well as in all other reef systems. Um, but yes, it's a bit hard for me to assess because it's one of those things when you see a reef, you can instantly come up with ideas about how to adapt things and better fit the process we know works elsewhere back into that context. So I think this is more the start of a conversation rather than a problem solving exercise immediately. Um, I guess the key message is that we're really happy to share with you the work that we've been doing, the successes we've had. And as Alex knows, I'm all about problem solving. So on every reef um, and on every reef trip, there are always unexpected problems, but we come up with solutions that allow this system to be quite adaptable. So even though the Caribbean system is very different and the Acropora groups there are tragically declined and smashed to the point where they're functionally extinct in the northern part of the Caribbean and the Florida Reef Tract, I still hold out hope that we can apply these methods to massively increasing the scale of larval production for those, as well as the massive corals that are really important in your reef systems. So exactly how to support the work you're doing. And I'm very aware of the work that you've been doing on restoration and really encourage you and applaud you for the work that you have been doing. But I think that trying to bring some of this approach in to support the work that you're doing is probably a sensible option. But until I know a little bit more and I get some more detailed information back from Alex now that she's seen the reefs and worked with you and understand the specifics of the problems you're facing from a logistical point of view, from a workflow point of view, and the sort of species that you have that you're going to need to focus on. Once that information's together, I think it's a case of just continually having some conversations, probably on a whole lot of video calls, until I could actually see your reefs and work with Alex and other people from our team to actually come over and try and contribute directly to the work you're doing.
Any other questions? We have another one in the back. Hi, this is Jennifer Keck again. I work at the Rosen Institute for Marine Sciences, which is hosting this conference, and I run the coral restoration program here. I'm just wondering what your feelings are about the sharing of gametes from country to country. Obviously we have such low genetic diversity in the species that exist, that there's even some concern that we might not have enough in a particular country. And given that these larvae are prone to genetic drift, and the chances that some of our larvae end up in Belize or Mexico, what are your feelings on if this is really going to, if we're really going to take this seriously and try to upscale coral restoration, that we need to start sharing that kind of genetic diversity, at least in regions these corals are.
Yeah, no, you've raised a really important issue, and I think the system is in such dire straits now that the only sensible approach is to rescue the genetic diversity that is still present by combining it with the genes of other populations. We know that the Caribbean system was, when it was, when most of it was healthy and the larval production was huge, that many of those larvae would naturally drift around to these systems, and humans have artificially put in place boundaries around nations, but the way in which broadcast spawners work doesn't work around boundaries made by humans. And so the only way to rescue some of these populations is to do exactly what your question was focused on, which is to take egg sperm bundles and gametes from different populations or adult breeding corals and put them somewhere where you can share the gene pools that are currently left in your remnant populations to massively increase genetic diversity. And the situation is now so desperate that if we don't do that within the next 5 years, we're going to lose so much more of the remaining genetic diversity. It's that urgent now, and it's absolutely essential to respect the ecology of the system rather than human artificial boundaries.
I just wanted to add to that. I found out that this week Andrew Bruckner from the University of Florida is actually in Taylor right now, and he has gotten permission from the Honduran government to take some actual living fragments of Acropora palmata. We have a population over in Taylor that is extremely thermally tolerant and survived this most recent bleaching event that wiped out a lot of our other corals. I'm really proud of our country, Honduras, for taking that initiative and giving the permission to bring that kind of genetic diversity somewhere else where they have the technology and the labs and most importantly the money and finances to really start exploring why these corals are so thermally tolerant and where to go with that.
That's absolutely wonderful news. This was obviously been a topic of discussion in the Caribbean for more than a decade now, and as the situation's got worse, the urgency has increased, and it was a very much a topic of conversation in the closing the coral cycle workshop that I was at in Arlington in the States back in February, because everyone recognizes the importance of this genetic exchange. It's what used to happen naturally, and now we've got to overcome the boundaries that people have put in place. So it's fantastic that Honduras has taken that step, because you are blessed with heat-tolerant Acropora survivors, and trying to mix that heat tolerance with remnant populations that will inevitably die unless more of the genetic exchange happens in other nations is absolutely essential. So I really applaud the Honduras government for enabling that, and for everyone who's been working in the background hard pushing for that for a long period of time. So that's a little bright spot that I'll take with me from this talk, because it's one of the most serious concerns that we have about the lack of process to enable sharing of the gene pools that are still present in the different reef systems. And yes, one of the things that I was most interested and most excited about when I thought that I might be able to go over there in person was to actually see some of your beautiful Acropora that's still on the reef.
Hello? I'm sorry, I'm an awful good question and answer, but as someone who's running a coral restoration program here, like one of my feelings is that as far as the, my fragmenting and not planning and having it be a little bit more costly and less scalable, I feel like that if every island nation had multiple nurseries where they have this genetic diversity in this really confined space, and even if they didn't have the manpower and the ability to outplant, just to have those mature corals reproducing once a year in these nurseries, and then having that higher chance of reproductive success is just something as well we should consider. I mean, because us, it's very inexpensive to go out and clean and maintain the nursery. It costs a little bit more to do the outplanting and the monitoring, but if we just had these sort of oases and nurseries of this thermally tolerant and disease-resistant and fast-growing genotypes all in one place, then these supercorals might get created just by having them there.
Yeah, and I totally agree. I totally agree with you. The Caribbean process is one of the areas where there is no alternative but to do more fragmentation and nurseries and cleverly combining the genotypes so that when spawning does occur, and if it's at a sufficiently coincident process, that you can get natural fertilization rates that are high enough to generate larvae, which will drift around onto other reefs. So I think in your context, it makes entire sense that you've got to increase spawning biomass, and the only way you can do that quickly is through some of the fragmentation and nursery processes. But I think the really important thing is that I think the situation's now so desperate that rather than relying on nature to take care of those larvae, I think every effort should be going into capturing the spawn from those nursery areas and, for example, either putting them into highly effective lab-based systems, which are expensive, but in some areas in the Caribbean, they are available and have proven to be very good at rearing larvae. But in areas where those aren't present, something like the larval pools would be perfect for rearing millions of larvae, and therefore you can control where those larvae are going to settle, either by settling them onto bits of dead coral or on limestone or devices, sea core devices or others, and therefore make maximum use of every larva that you generate out of all the hard work that you put into restoring those populations through fragmentation and nursery areas. So I think it's essential that that's been done, but I'd say the next stage is to don't rely on nature, because we haven't got every larvae is precious in the Caribbean particularly. So make sure you use those larvae as effectively as possible, I think, by retaining them and ensuring that as many as possible end up as settlers.
So if we have no more questions, I think we just have a last question over here. One more. Hey, Professor Harrison, it's Jazz.
Hi, how are you? Nice to see you.
Thank you so much for your presentation. Absolutely fascinating. And I just kind of wanted to pick up on a couple of topics I've heard spoken about today at the symposium. And just something else that I personally, as a non-scientist, found really interesting and important. I just wouldn't mind hearing you elaborate a bit more on. The first one is about zoning and how do we protect the work that the scientists are doing to restore coral. How does that work in Australia? What kind of initiatives have you seen be effective? We had a bit of discussion here on, how that's done here in Honduras. How can we improve those efforts to protect the work that's being done from tourism, fishing, all these different kind of threats to the science, so to speak, but also really important to industries here. The second one, something that just interests me a lot, on one of your slides, where for the work that you're doing in the Philippines, you had like $17, and you kind of, I think you were kind of getting to the cost effectiveness of the projects that you've been implementing. And I think that's incredibly important for sustainable development projects. And I just wanted to hear more about how you do that, how you conceptualize that, your achievements in that space. Thank you, Peter.
Yeah, thank you. No, they're both really important topics. So obviously, as I said earlier in the talk, active management of marine protected areas and remnant populations in particular of corals and the remnant healthy reef systems is absolutely essential to control human activities and stop extraction and things. On the Great Barrier Reef, I guess there's the luxury that 1/3 of the Great Barrier Reef is protected from fishing. So we know, and it is actively protected. So the government invests a lot of money by going out and having marine parks rangers and a whole range of other things. And everyone is so aware that when they see something that's illegal happening, they will report it really, really quickly. So there's this whole network now. The culture of people who use the Great Barrier Reef has changed to the point where everyone recognizes how important it is that those zoning plans are respected. Clearly, there's always people on the fringe that will try and disrespect them and take advantage, but they can be shut down pretty quickly. I guess the more serious issue is in some developing nations like the Philippines, where there are literally hundreds of marine parks that are declared, but a very, very small percentage of those actively managed. And so they are what we call paper parks in that they're gazetted and they sometimes have a management plan, but there's no funding or resources to enforce that process. And so that's the biggest concern. So in the Philippines, the approach we're taking is that we're only working in areas where we feel that the reef is recoverable. In other words, the original impacts such as blast fishing are no longer occurring, where the local community is aware of just how much they've lost and how desperate they are to get it back. And we actively involve the local community in the stakeholder training so that when our project funding finishes, we know that the local community already has a sense of purpose and ownership of the restored areas. So that's why we're working so much we're working with local fisher folk, we're working with local government units, we're working with anyone who will work with us, including school kids and, family members, community members, all of that, and right through to non-government organizations and tourism groups as well. We're very actively involved with tourism groups, particularly on the Great Barrier Reef. The scaling up of the Great Barrier Reef restoration is actually going to be managed through partnerships with tourism operators and traditional owner Aboriginal groups. That's how the scaling is going to occur in its greatest extent. So there's some really important things about making sure that the areas that you're restoring are going to be respected. And the best way of doing that is to involve the local community in that, and particularly the people involved with, tourism or management combined with local fisher folk and local community members, because they're the ones that need to be engaged so that when they've put effort into restoring a place, they're more likely to protect it in the future. The second point, excuse me, was really important about cost effectiveness. So in the early stages, in the first experiment that Dexter did during his PhD, we ended up with the breeding corals, the production cost of breeding corals being calculated at just over $20 per breeding corals. And so that was quite good. But it's nowhere near good enough to be scaled. So as we scaled up in that next experiment that I showed, where we tripled the amount of larvae, the cost production came down to $17 per breeding coral. And if you continue that process by increasing the scales of production, our aim is to get breeding coral costs down under $10 a breeding coral and eventually under $5 a breeding coral. And at that sort of cost structure, you can start to imagine effective restoration at multi-hectare to kilometre scales. Because if you look at the restoration costs of all restoration activities, including terrestrial activities around the world, they are very costly exercises. And in the case of rescuing reefs, there's no alternative but to spend money to make sure that you can restore reefs effectively, and therefore bringing that cost per unit of breeding coral down is a really important aspect of that. And that's why we're trying to simplify these processes and make them very adaptable to use by local communities, which brings the cost of the production of those larvae and therefore the settlers and therefore the juvenile corals way down to a reasonable level. Does that sort of answer the key parts of your question, Jazz?
I think so, unless anyone else has anything to add on to that. But I think it's important to mention those things considering.
Yep.
Oh yeah, yes. Thank you very much, Peter.
Yeah. Yeah, we're very mindful about making it like this is no longer just a research process, or it's research for management and restoration, and therefore it has to be cost effective, it has to be adaptable, and has to be able to be used by local communities and non-scientists. And that's exactly what we've ended up with. And so that's exactly what we're doing now in the Philippines, on the Great Barrier Reef, in the Maldives, and maybe next year in the Honduras. Okay.
Thank you very much, Dr. Harris. We see a little issue with the connection, but thank you so much for your insight and experience with us.
Okay. Thank you again for the invitation to talk. Thanks, Alex. We'll catch up sometime when you're back and recovered from jet lag again. I'm still jet lagged, badly jet lagged. And I thank again, Jazz, thank you for organizing this and the Australian government and the embassy staff who've been behind this to help with the process of getting Alex over there to start the conversation with the key people in the Honduras. Thank you. Okay.
Thank you so much, Dr. Harris.
Okay. Thanks. Bye now.
Remarks. Lástima que se me fue el doctor.